Assalamu alaikum. Hello, my dear students. How are you? I hope you are well by the grace of Allah. Today, we will learn about mitosis cell division from chapter 2. So, my dear students, our today's topic is definition of mitosis cell division, its characteristics, why mitosis is called, equational division, steps of mitosis cell division, importance of mitosis cell division, and results of irregular mitosis cell division. My dear students, definition of mitosis cell division. You know the process of cell division by which one mother cell divides into two daughter cells, where number of chromosomes and the characteristics of daughter cells are same as in mother cell, is known as mitosis cell division. My dear students, then where does mitosis takes place? My dear students, you know mitosis takes place in all body cells of plants and animals. It occurs in all meristematic cells of plants. For example, at the apex of root and stem and growing leaf. It also takes place for the formation of reproductive organ. Okay, my dear students, characteristics of mitosis cell division. It can take place in haploid as well as diploid cells. Both the daughter cells formed through mitosis received similar characters and number of chromosomes as that of mother cell. The original structure of the chromosomes remains unchanged in both the daughter nuclei. Okay, my dear students, stages of mitosis cell division. You know there are 4 stages of mitosis cell division: 1 prophase, metaphase, anaphase, telophase, and cytokinesis. My dear students, prophase. We are drawing prophase stage. At first, we have to make a rectangle shape. Prophase stage of plant cell. At first, we have to make rectangle shape, and outside the shape, we have to make another line. The inner line is cell membrane and the outer line is cell wall. Then inside the rectangle shape we have to make a circle. Then we make 4 chromosomes inside the circle, 2 long chromosomes and 2 short chromosomes. Then we make a nucleolus here. This is the nuclear membrane. This is the chromosome. This is the centromere. And this is the nucleolus. And this figure's name is prophase. States. My dear students, during early process, the chromatin network becomes visible as it condenses into separated threads for chromosomes. The nucleus become larger in this stage. Dehydration starts in the cell, especially in the chromosome. When stained, the chromosomes are visible under the microscope. By this continuous dehydration, chromosomes gradually become shorter and thicker. At this stage, each chromosome splits longitudinally but centromere remains intact. Each part of chromosome is known as chromatid. At the end of this stage, nucleolus and nuclear membrane disappeared. My dear students, now we are drawing metaphase stage. At first, we have to make two poles. This is one pole and this is another pole. Then we have to make the equatorial zone. This is the equatorial zone. Then we make some straight lines to attach the dots. Okay, then we make centromere, 4 centromere in the equatorial zone and make chromatids, thick chromatids of chromosomes. In this stage, chromosome become most short and thick. Okay, this is the equatorial zone. This is the pole. This is another pole. This is the chromosomal fiber. And this is the spindle fiber. Spindle fiber. And those are the chromatids of chromosome. This is the centromere. And this figure's name is metaphase. Stage. Metaphase stage. At this stage, spindle apparatus is developed from the two poles. The middle part of the cell is called equator. The fibers which are spreaded from one pole to the other is known as spindle fibers. The fiber attached to centromere of each chromosome is called traction fiber or chromosomal fiber. In animal cells, ester rays are radiated from the centriole present in the two poles. At this stage, chromosomes arrange at the equatorial zone. The centromere of each chromosome remains at the equator and the arms of the chromosomes remain towards the poles. The arrangement of chromosomes at the equator is known as metakinesis. At this stage, chromosomes become shorter and thicker. The two chromatids become clearly visible and the centromere divided into two so that the two chromatids can be separated. Anaphase stage. The two separated chromatids may called daughter chromosomes of each chromosome move towards the poles by traction fibers. During movement of the chromosomes, centromeres remain ahead while the arms followed them. My dear students, now we are drawing Anaphase stage. In this stage, centromere of chromosome completely splitted and make two daughter chromosomes, and two chromosomes moves to the opposite pole. So we have to make this type of structure here, spindle apparatus. Those are the spindle apparatus. Then we make centromere. And disappearing spindle apparatus. Thin and long chromosomes. Disappearing spindle apparatus. Now level. This is the pole. This is the daughter chromosomes. This is the daughter chromosome. This is the disappearing spindle apparatus. This is another pole. And this figure's name is Anaphase stage. In this stage, different types of chromosomes are to be seen, such as V-shaped, this is called metacentric chromosome, L-shaped, this is called submetacentric chromosome, J-shaped, this is called acrocentric chromosome, and I-shaped, this is called telocentric chromosome. These different shapes of chromosome are due to location of centromere. The anaphase stage comes to an end when the daughter chromosomes reach to the poles. Due to contraction of traction fiber and the expansion of the stem body. Then telophase stage. Now we are drawing telophase stage. We are drawing 4 chromosomes. Nucleolus, nuclear membrane. We are drawing 4 chromosomes here with a centromere and a nucleolus. Those are the spindle apparatus, disappearing spindle apparatus. At the last of this stage, they disappear completely, and at this stage, cell plate. Cell plate are formed. So this is the nuclear membrane. The nucleolus. And this is the chromosome. This is the cell plate. And this is the disappearing spindle apparatus. And this is the daughter. A complete nucleus. And this figure's name is telophase. The daughter nucleus take position in the two opposite poles. Hydration takes place in the chromosomes. As a result, chromosomes start expanding and gradually become thin and long. At the end of this stage, nuclear membrane reappears. A spindle apparatus gradually disappears. Nuclear membrane and nucleolus reappears. Okay, then cytokinesis. At the end of telophase, cytokinesis starts. At the equatorial region, minute particles secreted by the endoplasmic reticulum and phragmosomes are deposited and combined together to form plasma membrane. Cell wall is developed by the deposition of hemicellulose, pectin and other components on the plasma membrane by the formation of cell wall. And then the mother cell divides into two daughter cells. Importance of cell division. Number 1, physical growth. The number of cells increases by the process of mitosis in multicellular organisms. As a result of growth and development takes place in living organisms. Reproduction. Unicellular eukaryotic organisms reproduce by this process. For example, Navicula, Chlamydomonas. Then maintenance the equality of chromosomes. By this type of cell division, all the body cells bear the same number of chromosomes. Then vegetative reproduction. This cell division helps to reproduce vegetatively of an organism. Healing of injury. This process of cell division helps to heal up the injury. Maintaining qualitative stability. This process of cell division maintains qualitative stability in the living organisms. My dear students, results of irregular mitosis cell division. Mitosis is controlled by various internal and external factors of cell. If this control is defective for any reason, uncontrolled mitosis occurs. This causes tumors and cancer cells. A protein called p53 usually plays a role in preventing cell division. If it is defective, it loses control of the cell cycle. This causes cancer cells. This is one of the causes of cancer in humans. Some growth factors work in cell division. Cancer cells make their own growth factors, so they do not need a growth factor to division. Many cells die every day in a multicellular organisms. This is to be accomplished through cell division. Millions of cells die in human body every day. Cell death occurs in two ways. One is necrosis and the other is apoptosis. Necrosis is the death of cells due to lack of nutrients or damage caused by toxins. Apoptosis. This is the genetically controlled death of the cell. Some cells are not needed for the body, so they have to be destroyed. In this case of human embryos, fingers are attached with thin tissues. The 5 fingers are later separated by destruction of the middle tissue. The longer a cell survives, the more likely it is to be damaged, which can easily lead to cancer. So, they need to be destroyed and killed. This generally applies to the epithelial cells of our bladders and intestines because they are constantly exposed to high levels of toxins. Thank you, my dear students. And your homework is the leveled diagram of the steps of mitosis cell division. I hope you all submit your homework properly. Thank you to stay with us.